Welcome, students, to Module 4.2.1, Introduction to Biotechnology. Biotechnology may be defined as the manipulation of genes to benefit human purposes. Biotechnology, many people think, is a brand new occurrence. However, it has been really going on as long as humans have practiced agriculture, because the long-used form of biotechnology is nothing more than selective breeding by the process known as artificial selection. This is when we'll talk more about this in a moment, I think, or a little bit into the presentation. The definition of a form of biotechnology that most people associate with biotechnology is genetic engineering, actually cutting DNA from one organism and putting it into another, rearranging genes. And what makes this recently developed form of biotechnology possible is the fact that the DNA of all organisms is similar and is interchangeable because of or by human manipulation. I don't know if you remember that movie, Mission to Mars. It was kind of a cheesy movie, but there's one scene in there, and I remember it was an advertisement. That's why I think I remember it. And it has these two actors right here. There they are. And they say, "That looks like human DNA." And my response is yes, and it looks like mouse DNA and bacterial DNA and fungal DNA. It all has the backbones and the same four bases. It's all interchangeable. And that's what's so amazing and at the same time maybe somewhat sobering and frightening about biotechnology, is that there's nothing outside its scope. Even human DNA is interchangeable with that of other species. If we think about some of the potential applications of biotechnology, some of the big ones, of course, are agriculture and biomedicine. Those are the two big ones. Others, not as big, they don't spring to mind. There's a lot of money in agriculture and also in healthcare and medicine. But it could also be used, biotechnology can also be used in forensics, the chemical industry, for environmental remediation, pollution cleanup, and other applications as well. Now, I want you to think about the species that you interact with on a regular basis, and virtually all the species in the human world are genetically modified. So unless you spend a lot of time out in nature, you don't encounter a lot of natural species. If you're a bird watcher, if you hike, and then you do encounter many natural species. But an average person lives in a house that is landscaped with modified species. Our food is modified species. Our pets are modified species. So virtually in our cities, the vast majority of species have been genetically modified. Of course, the one exception is other human beings. And so to some, that would make humans the last frontier of biotechnology. Of course, biotechnology has a long way to go on other species. I'm just stating the fact that humans are widely modified. For the balance of this particular introductory module, I'm going to outline some of the tools that are used in biotechnology, and then we'll talk about the foremost tool of biotechnology, and then we'll end this module. But for right now, here's a list of some of the tools for manipulating DNA in the practice of biotechnology. The first one, restriction enzymes, is undoubtedly the biggest one, although they all interact and are used together in different techniques. DNA ligase is an important enzyme we'll learn about. Plasmids are important as vectors for genes. An enzyme called reverse transcriptase is important. And of course, the technique known as PCR, polymerase chain reaction, for amplifying DNA, taking one molecule and turning it into millions. Let's look first at restriction enzymes. These are enzymes that cut DNA at specific sites. The sites where a restriction enzyme cut is called a restriction site. And this is one of the big tools in biotechnology, because by being able to control where you cut in a particular gene, or in a particular DNA molecule, I should say, this can allow you to make recombinant molecules. Now, when we say recombinant DNA, we're meaning we are recombining it. We're cutting DNA from one source and putting it into another source, and that's recombinant DNA. Now, restriction enzymes occur in nature, and that's what's interesting. Humans have discovered and then appropriated these enzymes for our own use in biotechnology, but that's not where they originally occurred. They naturally occur in bacteria, and bacteria use them to combat bacteriophages. And you'll remember when we talked about the Hershey and Chase experiment that a bacteriophage is a bacteria-eating virus. So these are viruses. So remember, viruses inject their DNA into the bacterial cells with the intent of their DNA taking over that cell. But evolution, whenever there's a thrust, there's a parry, and that's sword fighting lingo for those of you that don't know what I'm talking about. But in any case, evolution responds to this attack by the development of restriction enzymes, and these restriction enzymes will cut up the DNA that comes in. And it's just interesting, and we don't understand why they cut at specific sites. And so that's kind of a cool thing. So let's talk about, there are hundreds of naturally occurring restriction enzymes, and people have actually made some artificial restriction enzymes as well, I think, but they're not widespread in use. How do restriction enzymes work? A restriction enzyme cuts at a specific site, so it has a specific sequence of bases, A's, G's, C's, and T's, and whenever the restriction enzyme encounters that, it will make a cut in a particular fashion there. And these restriction sites are typically palindromes. They read the same in the 5' to 3' direction as in the 3' to 5' direction. Like the word mom, it reads the same from both ends, or madam, I'm Adam, something like that, I suppose. And most restriction enzymes, but certainly not all, cut in an offset manner. So they don't cut straight across, and so they have this overhang on one of the two strands of DNA, and we call this a sticky end because it's hanging over, and it will readily make hydrogen bonds with a corresponding sequence on another DNA strand. And so sticky ends and offset cuts are very typical of the way that restriction enzymes cut. I will point out to you a second tool, that we'll just throw it in here, and that is that the enzyme DNA ligase. This is an enzyme that will actually make the covalent bonds between nucleotides, and so this will actually make the backbone. So initially, if two strands of DNA are held together by sticky ends, DNA ligase can go in there and make phosphodiester bonds in the backbones of DNA to make it a solid molecule. Let me show you, I guess right now it's kind of hard to envision this, so let's look at an actual example. This will kind of bring it home, I think. An example of a restriction enzyme is EcoR1. EcoR1, that is a Roman numeral 1 at the end, and the R stands for restriction enzyme. Eco stands for Escherichia coli. So this EcoR1 was the first restriction enzyme isolated from Escherichia coli. You might know Escherichia coli better as E. coli, as it's commonly known. So EcoR1 always cuts the sequence GAATTC, just like that. And so I've shown wherever I have a B, it's any base, but then I show you the restriction site, and it makes this offset cut, just like that. And so the overhang is going to be TTAA that's hanging over and not there when it's cut. So that's what we mean by sticky ends. And so here's a whole list of restriction enzymes. You don't need to memorize any of them, and this is actually showing where they cut, where they make the cut, and so this is the restriction sites. And notice that most of them have sticky ends, but here's one that cuts bluntly across right here, you can see. So there are some that don't make offset sticky end cuts, but most do. And so this is showing sticky ends, I guess, in a way. There's the sequence, and there we can see that EcoR1 would cut just like that, leaving TTAA as sticky ends. Now, this actually shows a recombinant DNA experiment using restriction enzymes. So DNA from two sources, green and yellow, two different organisms, but they both have the same sequence of bases. And so restriction enzymes, they really do allow us to cut DNA in just this way. So we actually call it, when we put a restriction enzyme with DNA, we call it a digestion. And so we digest the two sources of DNA and we get sticky ends, and some of them will come together, and we just add some DNA ligase and we can get new DNA, recombinant DNA that's never been made before. And so initially just the hydrogen bonds are holding the two fragments, but then DNA ligase is used there. And so this is another picture showing the same or a similar type experiment, cutting DNA from one source, the turquoise, and then from another, the purple, and then they come together. And they have little nicks there, and the DNA ligase actually fixes the nicks in the backbone, so there are none. And so a very common technique. Restriction enzyme digest followed by DNA ligase, and you can get new things forming there. And that's going to end this module. We'll end right there, and we'll resume talking about some of the other tools in the toolbox of biotechnology in our next module.